Have you ever wondered why humans are so drawn to alcohol? Is it just a cultural thing, or could there be a deeper, evolutionary reason? Well, a fascinating new study might just hold the answer, and it all starts with our closest living relatives, chimpanzees. Picture this. Deep in the forests of Uganda and the Ivory Coast, chimpanzees are going about their day, munching on ripe, juicy fruit. It sounds idyllic, right? But here's the twist, that fruit isn't just sweet, it's also slightly fermented. And that fermentation means it contains ethanol, the very same type of alcohol found in your beer or wine. For a long time, scientists have suspected that chimps might be consuming alcohol, but a recent study published in Science Advances is the first to really dig into the numbers. Researchers meticulously observed two different groups of chimpanzees, collecting samples of the exact same fruits they were eating. What they found is pretty astounding. On average, the fruit contained about 0.3% ethanol. Now, that might not sound like much, but consider this. An adult chimpanzee eats around 4.5 kilograms or 10 pounds of fruit every single day. When you do the math, that adds up to about 14 grams of pure ethanol consumed daily. To put that into perspective for us, 14 grams of ethanol is roughly the amount you'd find in 1.5 standard drinks. But here's where it gets even more interesting. When you adjust for the chimp's smaller body weight, their daily intake is equivalent to a human drinking about 2.5 alcoholic drinks every day. That's a substantial amount. The lead author of the study, Robert Dudley from UC Berkeley, points out that the chimps are eating 5 to 10% of their body weight in ripe fruit. So even those low concentrations of alcohol add up to a significant daily dose, and this is just a baseline. If the chimps are actively seeking out the ripest, most sugary, and therefore most fermented fruits, they could be consuming even more. This brings us to a really cool idea known as the drunken monkey hypothesis, a term coined by Dudley himself. The hypothesis suggests that our own human attraction to alcohol isn't a modern quirk. Instead, it might be an ancient inherited trait from our primate ancestors who learned to associate the smell of alcohol with ripe, energy-rich fruit. Think about it. In the wild, fruit is a crucial source of calories. The ripest fruit has the most sugar and is the most nutritious. It also happens to be the fruit that's starting to ferment and produce ethanol. So, over millions of years, primates might have evolved to be drawn to the smell of alcohol as a reliable signal that they've found a high-calorie meal. It's like a natural signpost pointing to the best food source available. The researchers behind this new study didn't just guess. They went into the field and sampled pulp from 20 different types of ripe fruit. They found that the most popular fruits on the chimp's menu, a type of fig in Uganda and a plum-like fruit in the Ivory Coast, consistently had the highest alcohol content. This suggests that the chimps might be deliberately choosing the boozier options, not just randomly munching on whatever they find. Now you might be wondering, are these chimps getting tipsy? Are there wild parties happening in the jungle canopy? Probably not. Kimberly Hawkins, a conservation scientist who wasn't involved in this study, explains that the chimps eat their fruit throughout the entire day. This slow, steady consumption means the alcohol is metabolized gradually, making it highly unlikely they'd ever feel drunk in the way a human would after a few quick shots. So, no need to worry about them making any bad decisions. The data confirms that a little bit of ethanol doesn't stop the chimps from eating the fruit, but the big question remains. Are they actively seeking it out? Is the alcohol an attractant? We can't say for sure just yet, but the evidence is mounting. When Dudley first proposed the drunken monkey hypothesis, it was met with some skepticism. But over the years, more and more studies, including this one, have provided evidence to support it. Primatologists have repeatedly observed monkeys and apes eating fermented fruit, and it seems this behavior isn't limited to just primates. Dudley notes that many fruit-eating and even nectar-feeding animals might use the scent of ethanol to find sugary food sources. So what does this all mean for us? It suggests that our relationship with alcohol is far more complex and has much deeper roots than we previously thought. It's not just about culture or social habits. It could be hardwired into our very biology, a relic from our fruit-eating ancestors. This research opens up a whole new way of looking at human alcohol consumption. It highlights the need for more research into why we're so drawn to alcohol and how this ancient evolutionary connection might influence modern behaviors, including alcohol abuse. Understanding our evolutionary past could be a key to better understanding ourselves in the present. So the next time you enjoy a glass of wine or a cold beer, you can think of our distant primate cousins foraging for fermented fruit in the forest. It's a fascinating link to our shared evolutionary history, a story that's millions of years in the making. What if I told you that in the hidden world beneath our feet, a queen exists who can give birth to two entirely different species? It sounds like something straight out of a science fiction movie, but for a tiny ant on the Iberian Peninsula, it's just another day at the office. Researchers have stumbled upon a survival strategy so bizarre it turns basic biology completely on its head. Get ready, because we're about to dive into the astonishing story of the Iberian harvester ant. Our story begins with a scientific puzzle. A team of researchers was investigating the Iberian harvester ant, or Messer ibericus, in the Mediterranean. Their initial data was strange. It looked like these ants were creating hybrid workers by mating with a completely different species, the Messer structor. Hybrids in the ant world aren't unheard of, but this case had a major red flag. The colony they were studying was on the island of Sicily. The problem? The nearest known population of Messer structor ants was over 600 miles away. That's a ridiculously long commute for a mating flight. Jonathan Romiguier, the lead researcher on the study, said they knew something was unusual, but they had no idea just how deep the rabbit hole went. This geographical paradox was the clue that kicked off a massive 5-year investigation. To crack the case, the team studied over 120 ant populations across Europe. They sequenced the genomes of hundreds of individual ants and even brought colonies back to the lab for observation. And what they saw was stranger than they could have ever imagined. From the eggs laid by a single Iberian harvester queen, 2 distinct types of ants emerged. One was hairy and the other was virtually hairless. The key visual difference between Messer ibericus and Messer structor. It was a biological bombshell. 1 mother, 2 species. So how is this even possible? The researchers discovered that the queen has a remarkable level of control over her offspring's destiny. It all depends on what the colony needs at that moment. If she needs to produce future queens of her own species, the Messer ibericus, she can. But the vast majority of the time, about 99% of her colony, isn't her own species at all. It's a hybrid workforce created through a process that is changing how we understand evolution itself. To understand this, we have to go back in time, about 5 million years ago. Messer ibericus and Messer structor were once the same species before they diverged. For a long time, they lived side by side in Europe. But at some point, the Iberian harvester ant queens lost the ability to produce their own workers. Why? The scientists suspect an evolutionary conflict, a kind of genetic civil war inside the colony, where a selfish gene pushed development towards making more queens, not sterile workers. This left the queens in a bind. A queen without workers is a queen without a kingdom. To survive, she needed a workforce, and the only option was to mate with the males of the other species. Messer structor. This strategy is known as sperm parasitism. The queen essentially borrows sperm from another species just to create her sterile hybrid workers. But this created a new problem. The queens were now completely dependent on finding males from another species, which is a risky and time-consuming business. Imagine having to track down a compatible partner from a different species every single generation, just to keep your colony from collapsing. There had to be a better way. And that's when the Iberian harvester ant evolved a strategy so audacious. It's never been seen before in any other animal. They didn't just borrow the sperm, they domesticated it. The study authors call it sexual domestication. The queen developed the ability to take the sperm from a mesostructure male and clone it. She could create a perfect copy of that male, entirely from his sperm, inside her own nest. Romiguier compared it to humanity domesticating livestock. The ants gained complete control over the reproduction of the males they once had to hunt for in the wild. This incredible evolutionary leap meant the Iberian harvester ants no longer needed to live near mesostructure populations. They could carry the genetic blueprint of the other species with them, stored within the queen herself. This allowed them to spread across the Mediterranean, creating massive colonies of invasive hybrid workers. They became a self-sufficient two-species system rolled into one. Researchers have even given this new mode of reproduction a name, xenoparity, from the Greek words for foreign and to give birth. It is the first ever documented case of a species needing to propagate another species' genome using its own eggs. The genetic analysis confirmed this wild scenario. When they sequenced the ant's DNA, they found that the only pure Mesor ibericus in the entire colony were the queens, and the males that could mate with them to produce future queens. Every single worker ant was a hybrid, a genetic mix of an Iberian queen and a structure male. But the most mind-blowing discovery was how the queen produced those Mesor structure males. She takes the sperm she has stored inside her body, and somehow she purges her own genetic material from an egg, allowing it to develop using only the father's DNA. These cloned males are almost perfect copies of the original Mesor structure father. They look a bit different, and they carry a tiny trace of the queen's mitochondrial DNA, but for all intents and purposes, they are clones. It's a natural cloning process that scientists are now desperate to understand. How does the queen's body know when to delete her own DNA? What is the exact mechanism that allows this to happen? Back in the lab, the research team witnessed this miracle with their own eyes. For 2 years, they monitored the artificial nests. Then, it happened. A single queen laid eggs that hatched into 2 different species of males. 3 were hairy, the classic Mesor ibericus. 2 were hairless, the cloned Mesor structure. It was the final, undeniable proof. This wasn't a theory. It was a living, breathing reality. The implications of this are huge. As one expert, Dr. Boomsma, pointed out, this isn't just a quirky biological trick, it's a powerful evolutionary advantage. By creating fitter hybrid workers, the Iberian harvester ant was able to expand its territory far beyond its original range. And once they were out of reach of the original Mesor structure males, evolving the ability to clone them was the masterstroke that stabilized the entire system. However, this strategy might also be its ultimate downfall. The system relies on cloning, which severely limits genetic diversity. In the long run, species that reproduce asexually or through cloning are often more vulnerable to diseases and environmental changes. Dr. Boomsma predicts that in a couple of million years, this fascinating ant will likely go extinct. But for now, it stands as one of the weirdest, most wonderful examples of evolution in action. The next great mystery is figuring out the how. How does the queen control the fate of each egg? How does she purge her own DNA? Unlocking the secrets of this natural cloning process could provide incredible insights for scientists trying to achieve artificial cloning in other species. The story of the Iberian harvester ant is far from over. It shows us that even in the most familiar of places, like an anthill in your backyard, nature is still full of breathtaking secrets waiting to be discovered. Ever since it was hunted to extinction 400 years ago, the dodo has been the ultimate symbol of a species lost forever. But what if forever isn't as final as we thought? A biotech company called Colossal Biosciences just announced what they're calling a pivotal step in their audacious plan to bring back the dodo. Get ready, because the story of de-extinction is getting wilder. So what's the big news? Colossal scientists have cracked a major puzzle. They've successfully grown specialized cells from the humble rock dove. Yep, the common city pigeon. These aren't just any cells, they're called primordial germ cells, or PGCs, which are the precursors to sperm and eggs. This is huge because the techniques that work for mammals, like cloning, don't work for birds. Birds develop inside an egg, making the whole process much trickier. This breakthrough with pigeon cells is like finding a master key. The team tried over 300 different recipes before finding the perfect mix to keep these crucial cells alive and growing. Now they're applying this same technique to the dodo's closest living relative, the beautiful Nicobar pigeon. The idea is to create a library of these germ cells, which will become the foundation for resurrecting the dodo. You might remember Colossal from their other ambitious projects. They've already announced the birth of dire wolf pups, and they're also working on bringing back the woolly mammoth and the Tasmanian tiger. This isn't just a handful of scientists in a lab. Colossal has raised over $500 million to fund this Jurassic Park-style flamboyant science, as one professor calls it. But how exactly do you go from a few cells in a dish to a walking, breathing dodo? The plan is complex and sounds like something straight out of science fiction. First, they'll take the PGCs from the Nicobar pigeon. Then, using DNA information from preserved dodo specimens in museums, they'll edit the genes of those cells to include dodo-specific traits. Here's where it gets even more interesting. They can't just grow a dodo in a lab. They need a surrogate mother. But instead of using a pigeon, they plan to use chickens. Why chickens? Because scientists already know how to genetically engineer them to be sterile, meaning they won't produce their own eggs or sperm. The next step is to inject the edited dodo-like Nicobar pigeon PGCs into the embryos of these sterile chicken roosters and hens. When these chickens grow up, they won't produce chicken sperm and eggs. Instead, they'll produce the dodo-like sperm and eggs from the cells they were given. When these chickens breed, the hope is that their offspring will hatch from the egg as a dodo-like bird. It is a multi-generational plan that the company's CEO says could take at least 5 to 7 years. Of course, this whole idea of de-extinction has its critics. Many scientists argue that you can't truly bring back an extinct animal. What Colossal is creating is a hybrid, a genetically modified proxy with dodo traits, not a 100% authentic dodo. An animal is more than just its genes. Its development is shaped by its parents, its environment, and countless other factors we can't replicate. Critics worry that calling this de-extinction is misleading and could distract from the urgent need to protect the species we still have. Colossal acknowledges this, stating their goal is to create a functional copy that looks and acts like a dodo, not a perfect genetic clone. They believe this new science has massive potential beyond just bringing back extinct icons. The same gene-editing technology could be a game-changer for modern conservation. Imagine being able to give endangered birds resistance to deadly diseases or help them adapt to changing environments. For example, scientists could find a gene for disease resistance in a museum specimen of an endangered bird, and then edit that gene back into the current population, making them stronger. This technology could give a crucial boost to species on the brink of extinction, like the pink pigeon, which is already benefiting from research linked to Colossal's work. So, while the idea of a living dodo walking the earth again is what grabs the headlines, the real immediate impact of this research might be in saving the countless bird species that are currently endangered. It's a powerful tool, but experts caution that it's not a magic bullet. Technology can't solve the biodiversity crisis on its own. We still need to focus on protecting habitats and preventing extinctions in the first place. But there's no denying that we are living in an incredible time. The line between extinction and existence is becoming blurrier than ever before. This pivotal step for the dodo is also a giant leap for genetic science. Whether it leads to a new flock of dodos or simply helps us save the birds we have left, it's a story that will continue to unfold in amazing ways. What do you think? Should we be trying to bring back extinct species like the dodo? Or should all our focus be on saving what's left? Let me know your thoughts in the comments below. These fluffy eucalyptus-munching marsupials are a symbol of the nation, but they're facing a silent and devastating threat that's pushing them closer to the brink. But there's a ray of hope on the horizon, a groundbreaking scientific achievement that could be the key to saving them. Stick around, because this is a story of survival, innovation, and a world-first effort to protect these incredible creatures. So what is this hidden enemy that's wreaking havoc on koala populations? It's a disease you might have heard of in a human context, but in koalas, it's a completely different and far more tragic story. We're talking about chlamydia. Chlamydia in koalas is caused by a bacteria called Chlamydia pecorum, and its effects are brutal. It leads to severe urinary tract infections, which are incredibly painful. It can cause blindness, making it impossible for them to navigate their treetop world and find food. And perhaps most devastatingly, it often results in infertility. For a species already struggling with declining numbers, a disease that stops them from reproducing is a catastrophic blow. Tragically, chlamydia is responsible for up to half of all koala deaths in the wild. Think about that for a moment. Half. It's a staggering number. In some areas, particularly along Australia's eastern coast in southeast Queensland and New South Wales, the situation is even more dire. Infection rates in certain koala colonies can be as high as 50%, and in some hotspots, it climbs to a terrifying 70%. Professor Peter Timms from the University of the Sunshine Coast has warned that some of these local populations are inching closer to extinction every single day. It's important to remember that chlamydia isn't the only threat koalas face. They were officially listed as an endangered species in Queensland, New South Wales, and the Australian Capital Territory in 2022. Their homes, the beautiful eucalyptus forests they rely on for food and shelter, are shrinking due to land clearing for agriculture and urban development. They're also vulnerable to attacks from dogs and getting hit by cars as their habitats become more fragmented. And then there are the bushfires. Recent years have seen unprecedented fire seasons in Australia, incinerating vast swathes of koala habitat and taking a terrible toll on the animals themselves. It's a multi-pronged assault on their very existence, and it's made the fight against diseases like chlamydia more urgent than ever. So how have we been fighting this disease until now? The primary treatment has been antibiotics. And while antibiotics can work, they come with a huge, often fatal side effect for koalas. Koalas have a highly specialized digestive system designed to break down tough, toxic eucalyptus leaves. The antibiotics used to treat chlamydia can completely disrupt the delicate balance of microbes in their gut, essentially destroying their ability to digest their only food source. So in a cruel twist, a koala could be cured of chlamydia only to starve to death because it can no longer eat. It's a terrible dilemma for veterinarians and conservationists. But after more than a decade of dedicated research, scientists at the University of the Sunshine Coast have developed something truly revolutionary: a koala chlamydia vaccine. And the even better news, it has just been approved for widespread rollout, a genuine world first in wildlife conservation. This isn't just a concept. This vaccine has already been put to the test. It's a single-dose shot that has been trialed on hundreds of koalas, both in captivity and, crucially, in the wild. The results are incredibly promising. A long-term study led by researcher Sam Phillips tracked wild koalas and found that the vaccine reduced the likelihood of them developing chlamydia symptoms by at least 65%. It also significantly decreased mortality from the disease. So how does it work? The vaccine is based on something called the major outer membrane protein, or MOMP, from the chlamydia bacteria. It provides 3 layers of protection. The first, it helps reduce the initial infection. Second, for koalas that do get infected, it helps prevent the infection from progressing to the severe clinical disease we talked about earlier, the blindness and infertility. And third, in some cases, it can even help reverse existing symptoms, giving already sick koalas a fighting chance. You might be wondering how the disease spreads among koalas in the first place. Much like in humans, it's primarily transmitted through mating, but there's another heartbreaking route of transmission, from mother to baby. A baby koala, known as a joey, can contract the disease from its mother while in the pouch. This means the disease can be passed down through generations, creating a vicious cycle that's hard to break. A vaccine that can protect breeding-age females is therefore a game changer, as it not only protects the individual, but also helps ensure the next generation is born healthy. The approval of this vaccine by the Australian Pesticides and Veterinary Medicines Authority is a massive milestone. It means it can now be produced and used widely, not just in small-scale trials. This is a critical tool that vets and wildlife rescue centres across the country can now add to their arsenal. It offers a proactive solution rather than a reactive and often risky treatment. Instead of waiting for a koala to get sick, they can be protected before they're ever exposed. This project is a testament to the power of long-term scientific research and collaboration. 10 years of tireless work by dedicated scientists has culminated in this moment. It is a powerful example of how science can provide tangible solutions to some of our planet's most pressing conservation challenges. Protecting koalas isn't just about saving a cute animal. It's about preserving a vital part of Australia's ecosystem and a global icon of natural heritage. This vaccine gives us a real reason to be optimistic about the future of koalas. While the challenges of habitat loss and climate change remain, tackling the devastating impact of chlamydia is a huge step in the right direction. It gives these resilient little marsupials a much better chance to survive, to breed, and to hopefully one day thrive in the wild once more. What do you guys think about this incredible scientific breakthrough? Are we living through a 6th mass extinction? It's a powerful, even terrifying idea. We've all heard it. Humans are the new asteroid, wiping out species at a rate not seen since the dinosaurs vanished 66 million years ago. It's a narrative of biological annihilation, a story of our own making. But what if that's not the full picture? What if the reality is more complex and, dare I say, a little more hopeful? A new study just published in the journal PLOS Biology is challenging this very idea. Now, before we go any further, let's be crystal clear. The study's authors and everyone involved in this conversation agree that biodiversity loss is real and it's a crisis. As study author John Weens from the University of Arizona puts it, every single one of these extinctions is a tragedy, and should never have happened. So this isn't about denying the problem. It's about defining its scale. The big question is, does the current crisis meet the scientific definition of a mass extinction? That's an incredibly high bar. A mass extinction event is typically defined as the loss of 75% of all species across the globe in a relatively short geological time frame. In Earth's 4.5 billion year history, this has only happened 5 times. This new research suggests we are not, in fact, at that level. So, how did the researchers come to this conclusion? Instead of just looking at individual species, they analyzed extinctions at a broader level, the genus level. Think of it like this. The genus Canis includes dogs, wolves, and coyotes. Losing an entire genus is a much bigger deal evolutionarily than losing a single species. The researchers, John Wiens and Kristen Sabin, dug through data on over 163,000 plant and animal species. They found that since the year 1500, about 102 genera have gone extinct. That's 90 animal and 12 plant genera. To put that into perspective, they analyzed over 22,000 genera in total. The extinctions represent less than 0.5% of that. The study argues that these losses, while tragic, are relatively rare and don't signal a global collapse on the scale of a mass extinction. Let's look at some of the famous examples of what we've lost. The dodo, which belonged to the genus Rhesus, is gone. The massive sea cow from the genus Hydrodamallus is gone. An entire family of Hawaiian honey-eater birds, the Mohoidae, has vanished. These are significant losses to the tree of life. The study also found a fascinating pattern. The vast majority of these extinctions, especially among birds and mammals, happened on islands. Places like Hawaii, New Zealand, and the Mascarene Islands were hotbeds for extinction. Why? Island ecosystems are incredibly fragile. When humans arrived, they brought invasive species like rats and cats, which native wildlife had no defense against. The study suggests that these island extinctions, while devastating, might not be representative of what's happening on a global continental scale. And here's the most surprising part of the research. The rate of these genus-level extinctions may actually be slowing down. The analysis found that the peak extinction rates weren't in our recent memory, but rather back in the 1870s, 1890s, and 1900s. The authors suggest that this slowdown could be, in part, thanks to modern conservation efforts kicking into high gear, especially for well-studied groups like mammals and birds. Now, this research doesn't exist in a vacuum. It directly contradicts a 2023 study by renowned scientists Gerardo Ceballos and Paul Ehrlich. Their work, which focused on 5,400 vertebrate species, concluded that genus extinctions were rapidly accelerating and that we are, without a doubt, in the 6th mass extinction. They argue that thousands of species we've lost in the last century would have taken thousands of years to disappear naturally. So who's right? The debate is fierce. The authors of the new study point out that the 2023 paper focused only on a small fraction of life, vertebrates, and excluded insects, plants, and fish. In contrast, their new study included a much wider range of life forms. This brings us to a crucial distinction that many scientists are now making: the difference between the biodiversity crisis and a 6th mass extinction. Everyone agrees we are in a biodiversity crisis. We are losing species and populations at an alarming, unnatural rate. This threatens ecosystems and even our own well-being. As Conrad Labandera, a scientist at the Smithsonian, puts it, the current biodiversity crisis exists, whereas the 6th mass extinction is interpretative. So why does this distinction even matter? Isn't it just semantics? Well, not really. Sadia Qureshi, a science historian, points out that framing the issue as an unavoidable apocalyptic mass extinction can backfire. It can make people feel hopeless, as if there's nothing they can do. If the end is already here, why bother trying to save anything? But if we frame it as a severe but manageable crisis, it changes the narrative. It becomes a call to action, not a eulogy. It empowers us. It reminds us that our conservation efforts can and do make a difference. We can still make a meaningful impact. One major challenge in all of this is simply knowing what we're losing. Confirming an extinction is incredibly difficult. You have to be certain the last individual of a species is gone. This is much harder for poorly studied groups like insects, fungi, and deep-sea creatures than it is for, say, a tiger. The IUCN data, which these studies rely on, has its limits, especially for the vast, unexplored world of invertebrates. Ultimately, whether we call it a sixth mass extinction or not, might be a distraction from the main point. As Stuart Butchert from BirdLife International says, the current extinction rates are already a grave concern. He also reminds us that mass extinctions happen over tens of thousands or even millions of years. We are living on a human timescale, making it almost impossible to know if we are at the very beginning of such a catastrophic event. What we do know is that the web of life is fraying. And we know that we have the power to either keep pulling the threads or start repairing the damage. The debate over the sixth mass extinction highlights the complexity of the problem, but it shouldn't overshadow the consensus. We are in a biodiversity crisis, and it demands urgent action. The hope lies in knowing that our actions matter, that conservation works, and that it's not too late to change course.